Okay, so welcome back to this session. My name is Zvonimir for the youngest, and now I'm going to chair the session, and is Zvonimir Dogic from a wonderful place from Santa Barbara that I know very well. I did my early postdoc here. Anyway, and he's talking chirality, entanglement, and allosteric mechanics.
Thank you very much. Again, thank you all for being here. Thank you for the kind invitation to come to Japan again. It's been very nice already, and I'm sure it will continue to be a very nice trip. I want to tell you a little bit about a particular experiment that that we are working on right now. So I'm an experimental physicist in soft matter. And being an experimental physicist, in many ways, I think is like being a farmer. You, as an experimental physicist, you plant these seeds, you try to grow a seed into a flower or a nice plant. Oftentimes, nothing happens. Sometimes things take a really long time and then suddenly start moving. And being in California, sometimes there's no rain, but it's been rainy the past 2 years, so a lot of plants are growing. And so this is one of the plants that I think is about to grow. But the seed was planted many, many years ago in my mind. I did my undergraduate studies at Brandeis University, and I had a great, I didn't realize it at that time. This is not working to advance. Okay, I had the great, privilege of being an undergrad student when there were two really great scientists there. One was Bob Meyer, who unfortunately passed away very recently, and the other one was Don Casper. He was a structural biologist. I talked to him in 1994, and he said, and the idea that we were always interested in soft matter is, a lot of proteins are chiral, and in liquid crystal, sometimes it manifests itself in a macroscopic chiral phase. And this was and it's still a mystery. And, I didn't talk to Don Casper a lot, but, probably 30 years ago, he said, well, if you want to understand chirality in liquid crystals, what you should really study is bacterial flagella. And so that we've tried that a few times, we failed. And, once I moved to Santa Barbara, I thought, let's try again. And I think there's some interesting results there, right? And so let me just orient you again. This is a bacteria that lives in various organisms and ponds. This is the cell body. This cell body has this chiral flagella. This is a helical filament. This is few microns. This is 8 to 10 microns. And the bacteria spins this flagella. There's a motor attached at its base. It spins at tens of hertz, and that propels the bacteria forward. If you look at a flagella, what you will find out if you do structural biology on the flagella, it's a hollow cylinder. It has a hole down the middle, and it has 11 protofilaments. Each protofilament is assembled from a monomeric protein called flagellin, and it's just a row of filaments, and then you have 11 of these rows of filaments, and you assemble this cylindrical structure that can again be many microns in length. And this is something that I told you already, and this is, for example, a movie of bacteria and being propelled by the flagella and acquiring motility. What we do, and this is certainly not us, we are not the first ones to do this, this is again a fluorescence microscope of a bacterial body with a fluorescently stained flagella. You grow a lot of this bacteria, and then you just shake it. You can't shake it too violently, because then you'll fracture the cell body and you'll get all kinds of junk in your solution. You can't shake it too little, because then you won't separate the flagella. But so if you shake it just right, you can separate the flagella from the cell body, and you get nice helical filaments, and this is the distribution of filaments that we assemble, and it's of the order of 5 microns or so. Okay.
So are you breaking up the filaments?
It's coming up. You know, and this took us a while to figure out. It's biochemistry, you know, things can go wrong. So let me just tell you a little bit why I think flagella are interesting. Again, they are assembled from flagellin monomers. Each flagellin monomer can exist in 2 conformational states. So they're about 5 nanometers, and the 2 states are different by 1 angstrom in size. And they have different bonding rules about how these protofilaments come together to form this how these protofilaments come together to bond, to form flagella. So you can have all of the conformational states of all the flagellin monomers be in an L state, and then you'll just get this helical flagella. In this case, it's a straight rod, right? And you can have them all in R state, and you'll also get a helical flagella that's in a straight rod. And this, by the way, was really a very pioneering work. This was mostly figured out by Sho Asakura, who also had a big influence for me because he's elucidated the depletion interaction, which we use a lot. And then that was followed up by a professor who was in Osaka, Keiichi Namba. So biologists really figured this out. But what can happen is that you can have, and in biology, you can have 11 different conformational states. You can have all of the protofilaments being in L state, all of the protofilaments being in R state, but you can have also a configuration. This is wild type flagella, where 9 protofilaments are in L state and 2 are in R state, right? And then you get incommensurability of the bonding interactions along the protofilament, and the way you resolve this is you create this helical structure, right? And you can, in principle, form any of these 11 helical structures. And so this is a pretty interesting feature, because now not only can we make chiral filaments, we can also make straight filaments, and we can, some of them are left-handed chirality, some of them are right-handed chirality. This is a, yes?
Sorry, is the 9-2 are the 2 straight ones next to each other?
Yes, they are always, all of the protofilaments in the same conformational states are next to each other. So you'll never have an LR, right? And that's just the structure that minimizes the elastic energy. And you know, you can create mutants where you mutate a single amino acid, and you can create various helical states. So for example, this is something we've isolated in our lab. This is the straight flagella. This is a normal flagella. It's a helix. This actually, by the way, is what you would find in a wild type bacteria, and this optimizes the swimming efficiency, right? And then you can have a curly flagella that has a smaller pitch, smaller diameter. You can have semicoiled flagella. So these are some of the numbers to be aware of, right? The hard core diameter of the flagella is 20 nanometers. They are very, very stiff filaments. They are 1 millimeter in stiffness, and they can have a helical diameter. That means the helical diameter is the diameter of the helix that varies from 0 to 1.5 microns. This is the most coiled flagella. This is, again, this filament over here, but it's blurred by optical microscopy. And so, for example, you can purify this flagella, and you can see that this is actually a very floppy thing, right? And this is not so surprising if you play around, you know, if you have kids, your kids, you know, like to play around with these kind of molecular slinkies, right? And slinky is very, you know, bendable, but if you take the material that a slinky is made out of, it's much stiffer, right? And so, you know, in a certain way, by using flagella, you can create a very, very soft molecular slinky. Something we spend a lot of time on and works finally now after several years of attempts is you can harvest the flagella. Again, they are all made out of the, yes?
Just one question about what you showed before about that more microscopic structure. Quite a bit earlier. You showed that cross section with the 11 units.
All right, I'm going. Tell me when to stop.
One more, I think, sorry. Okay, here. So when you said there's some fraction of left-handed, some fraction of right-handed?
yes.
So you would see that also at this more microscopic level, or?
Yes. Like this is one protofilament. This is a bunch of flagellin monomers that come together in this configuration. And all of these filaments, there's 20,000 filaments, are either in L or R state. It's extremely cooperative.
So all of them would be in this.
Yes.
Thanks.
Okay, let me go back, right? So what you can do is you can take flagella, you can, heat it up, that breaks up the flagella into monomers. And then with monomers, what you can do is you can make some seeds by putting in a lot of chemicals. You create short seeds, you mix short seeds with monomers, and you can repolymerize the flagella. And so not only can you control the helicity of the material, you can control the length of the material, right? So this is a harvested flagella 8 microns, and you can repolymerize it, and you can get these beautiful filaments that are 50, 100 microns long, right? So this really is, a molecular spring, right? Physicists always think about springs that are virtual. This is a real spring, right? And it has several interesting properties, right? So this is my talk outline, right? Is I've told you a little bit, some facts about bacterial flagella. I'll tell you what happens when you take a lot of this flagella and just concentrate it. What are the properties of the suspension of the flagella? And then at the end, I'll tell you some rather unique features is that engineering these filaments have unique polymorphic transitions between multiple states. So something we've noticed very quickly is you can take flagella, straight flagella, and you can see this is a kind of real poor man's rheology. This flows as easily as water. And then you take helical flagella and you say this definitely has a very different viscoelastic response, right? And so again, this is straight flagella, this is helical flagella. So let me just give you a minute introduction of how we actually characterize that. What you define is you define something that is viscoelastic coefficient, elastic coefficient, sorry. So, in one limit, you can have purely elastic materials, that's just your spring. On the other limit, you can have viscous material that flows with some viscosity, but complex materials are going to have, behave both viscous and elastic, right? And so this is telling you what is the frequency of deformation. And so if you have a polymer network, for example, at certain time scales, if you apply a deformation, the polymer is going to pull back on you, right? But if you apply that deformation on a much longer time scale, the polymer can relax through what's called a reptation mechanism. And that what happens is then the viscous modulus dominates. Again, you have G prime that tells you what is the elasticity of your material. And you have G double prime that tells you what is the viscosity of your material. So in this regime, G prime elasticity is larger than G double prime. And then in long time scales, because the sample disentangles, that reverses. You can change that by simply cross-linking your material, and then you just simply have a bunch of springs that just sits there. And so material will always relax on all time scales. So if you take a rheology and you measure rheology of straight filaments, what you'll see is, this is loss modulus. And so for straight filaments, the loss modulus is always much larger than the elastic modulus. So this is more or less a viscous material. On the other hand, if you take a helical polymer, what you'll see is that over a certain frequency range, the storage modulus dominates the loss modulus. And so this is telling you that you have a viscoelastic polymer solution. Okay? You can, control the concentration of these helical filaments. For example, now we are tuning the concentration of helical filaments at 1 mg per mil. Again, open circles are loss modulus, the viscosity dominates. We increase the concentration. You see at 2.5 mg, you obtain a regime of frequency where the elastic modulus dominates. And if you increase the concentration, this is more or less an elastic material over all length scale and time scales. And so that's telling you something about what are the microscopic motions of these filaments, right? And so to understand that, what we did is we fluorescently label 1 out of 10,000 filaments, and we were interested in what is the microscopic dynamics of straight filaments and what is the microscopic dynamics of helical filaments. This is one filament, but you have to remember that there's many filaments that are unlabeled, right? If there's one filament, it would just exhibit rotational and translational diffusion as was predicted many years ago, right? But if you have a helical, if you have many filaments, what essentially happens is that this filament is caged, right? This is still a very low volume fraction of less than 1%. And so this filament is free to diffuse along its long axis, but if it translates or rotates, it very quickly hits a cage. And so you can define this kind of cage that was, talked about by Edwards and Masao Doi many years ago, right? A curious side note, I've worked with biopolymers. If you look at these biopolymers, usually you fluorescently label them, you look at them for 5 minutes, you photo bleach the sample. We can fluorescently label this with rhodamine, and these things remain stable for 4 or 5 days. We are looking at this with confocal microscope. There is no bleaching whatsoever. I have no idea why that is. We are very happy that that's the case. But it's a curious observation that I would love to talk to some photochemist about. Again, this is the position of the filament as a function of time. You can see it goes back and forth, and on a long time scale, it starts changing direction. So this is the mean square displacement as a function of time. And you can see that this is the displacement along the filament. The filament moves along its long axis. And you can see it's more or less a linear diffusion. And if you look at its orientation, this would be the sloped one, right? That would just be the bead moving in a viscous solution, would have a mean square displacement that increases linearly with time. You can see that the orientation is, subdiffusive, right? Because this thing is effectively caged. So this all makes sense, one can really do quantitative experiments here. We are tracking these filaments in 3D over 3 days and getting really very nice quantitative data, and those are the experiments in progress. But now let's look at helical filaments and what is the motion of helical filaments and how it is different than motion of straight filaments. So this is a single helical filament, and you can observe something, it's diffusing, there's many other unlabeled helical filaments, and you can observe something very curious, right? This is a helix, but the microscope has a limited observation, so it just looks like a sine function, and you can see there is a peak and a valley here, right? And what do the peaks and the valley means, right? That means that, if I had a helix, and if I was just moving it, translating it, that would mean that the peaks and the valleys of the helix would move and translate with the center of mass, right? But here you can see there is this peak, and this peak stays always at the same position, okay? You can again observe that here, this is a time lapse, there is a valley here, and you can observe this thing is fluctuating along its long direction, and you can see that always this valley over here stays constant, right? The third way you can do it is superimpose a bunch of images. And again, you can see that over several minutes, again, there's the peaks and the valleys, and they always stay the same. And so this is indicative of really chiral microscopic dynamics, right? What is really happening here is that you have an entangled filament, and this filament cannot just simply translate. It cannot simply rotate. It can do what we call chiral corkscrewing dynamics. It's very similar to, if you want to open an old-fashioned bottle of wine, right? If you have a corkscrew opener, you have to rotate it, and every time that you rotate it by 2 pi, it translates by a helical pitch. And you can really see that over here, right? That this really is exhibiting this corkscrew dynamics that is very constrained. And that's not so surprising. Again, you have to think about the dimensions, right? This is greatly exaggerated, but this is your tube now, right? Now you're no longer confined to a straight tube. A helical filament is confined to a helical tube, right? The diameter of the helix is 20 nanometers, and the hardcore diameter, but the diameter of the helix, the diameter of the filament is 20 nanometers. The diameter of the helix is about a micron, and the mesh size of the network is much less than a micron. Yes.
Does it make a difference whether the network itself is chiral?
I don't know, that we have not done that experiment yet.
But what you showed us was straight background? or-
No, this is the helical background. Yes, yes. Helical background again is much more entangled, right? So the straight, there could be difference, but I don't think in principle, I think I could put this into any network and it would exhibit the same structure, right? And so what's happening is I can't pull this thing. If I just translate this thing, this helix is going to bump into all other filaments. On the other hand, if I just purely rotate it, that can happen. But if I do this corkscrewing dynamics, then the system is free to relax. And again, you can measure the diffusion along the length along the filament length, and the rotational diffusion, and this is diffusive and this is subdiffusive. Now, what you can ask is an interesting question, right? Is what we really, this. Oops, what's going on? It's no longer working. No, there's battery, but
Fine.
oh, is it working? I'm just not pressing it right. I don't know how I'm gonna. Okay, so I don't know. Okay, an experiment a cute experiment that would be really kind of interesting is if you could prepare a helix that half of it is left-handed, and that part of the helix would want to do left-handed rotation, and then you could switch to a other helix that's a right-handed, and that helix would want the whole thing to rotate in a different directionality, right? Now that cannot obviously happen, right? And so the whole thing would just be completely jammed, right? That is a thought experiment. Unfortunately,